This is a production of Cornell University.
Awesome. Thank you, Savannah. So as she said, my name is George Stack, and I'm a second year, and I'm going to be talking about a binary choice network bioassay to assess aphid preference in hemp. So just to see if I can get my slides to advance, just to introduce the study system. Hemp is cannabis sativa with less than 0.3% THC. It has many post-harvest uses, including for fiber, for grain, and for cannabinoid production like CBD. And a major insect pest of hemp is the cannabis aphid. It's a specialist, and it feeds on hemp and can be a problem in controlled environments and field settings. So if we're thinking about breeding plants for resistance to aphids or any other insect pests, we can think about three different categories of insect resistance. One is antibiosis, which disrupts the biology of the insect. One is antizoonosis, which disrupts the behavior of the insect. And the last is tolerance, which involves incorporating traits that allow plants to withstand and recover from insect damage. So I'll just be focusing on one of these today, antizoonosis, particularly aphid preference. So if we want to assess insect preference for many genotypes, this can be kind of a challenging problem. One way that we might do this is by doing a simultaneous comparison of all of the genotypes that we're interested in. So in this example here, let me throw my pointer on. We've got 5 different genotypes represented by the 5 different colored circles. We can put them all in a cage, put a bunch of insects in there, and look at how the insects distribute themselves among these plants and use that distribution as a metric for insect preference. We could also consider comparing all of our genotypes to a control genotype. So in this design, each genotype is compared to this gray control genotype, and based on how the insects distribute themselves among these 2 plants, we can gauge preference relative to the control. So both of these designs have pros and cons. One con of the simultaneous comparison is that once you get very large numbers of genotypes, say maybe 10 or 20, then the insects can't adequately assess all of the genotypes and then pick their favorite. And one con of the comparing all genotypes to control is that the results are very conditional based on what this control genotype is. You can imagine if there was a different control genotype, then you might get a different result. So I was wondering if we could improve on that. And instead of comparing all of our genotypes to a control genotype, could we instead pair each genotype with multiple other genotypes? So take, for example, if we had 35 genotypes that we were interested in, if we wanted to make all pairwise comparisons, that would be almost 600 pairings, which is a lot of pairings, arguably too many pairings. So then my kind of secondary question is, can we use less comparisons? And based on the comparisons that we do make, can we infer information about the comparisons we don't make? Okay, so if we want to do less comparisons, is there an optimal reduced set of comparisons that we can make that will give us our, the best ability to do this inference for the comparisons that we didn't make? So to try to answer the second and third questions there, I used network modeling to try to optimize an experimental design. So I modeled the entire experiment as this network where each node in the network is a genotype, and each connection between 2 nodes is a bioassay between 2 genotypes. In parameterizing the network, I made sure that each node was connected to 10 other nodes, so each genotype is compared to 10 other genotypes. And in the optimization, I wanted to minimize the degrees of separation between any 2 nodes. So in this example here, we have our focal node. The cream colored nodes are 1 degree away, the yellow are 2 degrees away, and the blue is 3 degrees away. So I figured by minimizing the degrees of separation between 2 nodes, that would maximize the ability for the model to do inference. So it turns out that there's about 280,000 different ways to build this network. And this is the optimal network. It's the same network, just organized in 2 different ways. And it's pretty nice mathematically, because in this network, every node is connected to every other node, either in a first degree connection or a second degree connection. So a high degree of connectivity. So we've got this design. And we can imagine, running this experiment, doing all 175 comparisons here, getting, say, aphid count data. But how are we going to translate that aphid count data into a metric for insect preference? And I tried to do that by using a model called the Bradley-Terry model. It's kind of interesting. It's a model that's often used in ranking sports teams. You might be able to see the parallels between, a sports team being like a genotype and a game between two teams being like a bioassay between two genotypes. In any case, in this model, each genotype gets an ability score or a preference score, gamma, and the outcome of a pairing is predicted by the ratio of ability scores. So in the simplest model, the probability that genotype I is preferred to genotype J is gamma I over gamma I plus gamma J. Quick example, if gamma I is 1 and gamma J is 9, genotype J is preferred, and we'd expect 90% of the insects to prefer genotype J and 10% to prefer genotype I. So I've got this model now, but before I want to run this kind of large bioassay in real life, I wanted to see if it would work in simulation. So to simulate the bioassay, what I did is I generated a set of true ability scores, so like true gammas, and then I used those ability scores to simulate count data for my network design. Then I fed that count data to an EM algorithm to estimate back my ability scores, and I correlated my estimated scores to true scores. So I was interested in seeing if varying the standard deviation of these true ability scores affected my model's ability to predict back the scores. And sure enough, it did. When there's a very low amount of variation in the true ability score, the model is not very good at predicting them back. But as you increase the amount of variation in ability scores, so variation in real insect preference, then the model becomes much more robust at making these predictions. So that's great. I was also interested in asking in simulation, is this network design actually better than the all-to-control design that I talked about before? So I generated some data, and I either used the all-to-control design to try to predict back my preference scores, or the network design to predict back my preference scores. And the network design consistently outperformed the all-to-control design, especially at high levels of variance for the true preference scores. So this was very reassuring, and I decided to move on to do a small pilot bioassay. So I selected 10 different high cannabinoid genotypes, and I designed a network where each genotype was compared to 4 other genotypes. I introduced 20 aphids on a piece of filter tip paper between the leaves, and I counted the aphids every 24 hours. And this is the data we got. It's a lot, so I'll kind of walk you through it. On the diagonal here, we have our 10 different genotypes. And at the intersection of 2 genotypes, there's a pie chart that shows the distribution of aphids on those 2 leaves. So for example, here we've got Sweet Caroline compared to 191067. Maybe it's 60% of the aphids were on Sweet Caroline and 40 were on 191067. Also, underneath each genotype, I have the preference score that was derived from the model using this count data. So those scores range from 3.5 down to 0.26. And now that we have these kind of estimated preference or ability scores, we can look at what we would expect the proportions to be, and then compare that to these observed proportions. So these are our kind of predicted proportions based on the preference scores from the model. And we can compare these to our observed proportions. So you can see some are very similar. Again, the Sweet Caroline 191067 comparison, maybe 60% compared to 65%. So pretty similar. Some are pretty different. This is a tangerine by TJ CBD. So not very similar, but generally they're pretty similar. So that's promising. So just quickly, some future directions. I'd like to replicate the experiment to see if the results are consistent. I'd like to compare preference bioassays, which is what I've been talking about today, to performance or no choice bioassays and see how those are related. And I'd like to optimize networks for other numbers of genotypes in comparison. So the largest I talked about today was 35 genotypes, 10 comparisons. But what if we wanted to do 100 genotypes? What would that look like? And I'll quickly say thank you to all the people who contributed to this work, the current and former members of the L Smart Lab, the whole Cornell Hemp team, and a big shout out to Cornell Stats Consulting, especially Erica and Joe for their help on the stats. We got some funding from New York State Ag Markets, and there's my contact info. I'm happy to answer any questions.
And so I guess I have a question. If you do in vitro assays with cannabinoids, how sensitive are the aphids to the cannabinoids? Is that really what's limiting their growth?
I don't think anybody has looked at that yet. I'm pretty interested in looking at the difference between insect performance with cannabinoids. We have some cannabinoid-free and cannabinoid-competent plants that I'd like to compare. I know somebody has done a diet bioassay with fall armyworm where they added cannabinoids to the diet, and they saw that the caterpillars performed worse on the diet with cannabinoids than the diet, the control diet.
Yeah, but you said this is a specialized aphid for hemp, so it may be highly resistant to the cannabinoids that have inherent.
This is true. This is true. So, I mean, I thought it was pretty interesting that, in these 10 genotypes that were all cannabinoid competent, there still seemed to be preference for the different aphid or the different genotypes for the aphids.
There is another question by Ed Buckler in the chat. How does this compare to the tricot design in terms of power? Tricot is used in participatory breeder evaluations.
Yeah, that's a great question. I have not looked at how this would compare to the tricot design and power. I would imagine that the power is very dependent on how many comparisons you make and how that compares to the number of genotypes. But that would be an interesting thing to look at, definitely.
We have clapping. So John-Luc, do you have a?
So yeah, maybe I'll put in a question here. So the approach of doing this network analysis for optimizing the experimental design is really great. And I wonder if you compared it to more traditional, I guess, approaches for experimental design, like alpha designs, or like every pair here is an incomplete block in a certain way. So yeah, any thoughts there?
I mean, yeah, great. I mean, it's a great thing to pursue. I haven't really looked beyond the comparing it to the Alta control design, but that's definitely something that I'd like to do in the future, and find out what is this really optimal, or are there other designs that could be more optimal?
Of course, the comparison to control is bound to be fairly suboptimal, right? I mean, it just seems like a whole lot of work, and you get a ton of information about the control. And anyway, so yeah, given how little this network design improved over the comparison to control, I wonder if lots of different experimental designs would be sort of similar.
Yeah, definitely. That's definitely possible.
Yeah.
We have one more quick question from Kevin Ahern. If you find a pedigree that appears resistant or tolerant, do you have plans to go after the underlying gene contributing to the trait? Would you perform GWAS?
I think there's definitely potential for that. I'd like to kind of look at, again, preference and performance across some diverse gene types. I've only looked at 10 so far to see what the variation is. Maybe there's not actually a lot of variation because the aphids are specialists, but or maybe there is, and maybe there's potential to do GWAS or a biparental QTL mapping population or something like that.
Great. Unfortunately, we don't have time for any more questions. There is another question and a comment in the chat, George, you might want to look at later, but thank you for a wonderful talk. It was great, George, and on to our next speaker, who will be introduced by Anna.
This has been a production of Cornell University, on the web at cornell.edu.